I'm Dylan.
Stain.
James.
So we're doing lesson 6 group problems. The first one has to do with Niemann-Pick disease. And we're talking about the frequency of the D allele. So it's a 1 in 150,000 individuals. So that would be our D squared value, which meant you have to take the square root of that to get D, which is 0.0025 or 26, and then subtract that from 1 to get your D dominant allele. For part C, you just square that in order to get your homozygotes. And then in part D, it's 2 times each of the allele frequencies, which gives you that as your heterozygotes. For 2, it was fill in the blanks, and it says the term in the Hardy-Weinberg equation that represents the frequency of the IB allele is Q, while the term in the Hardy-Weinberg equation that represents the frequency of the IA-IB genotype is 2PQ. For B, DiGeorge syndrome is an autosomal dominant disorder that affects 1 in 4,000 individuals in the human population. The 1 in 4,000 individuals in the population affected by DiGeorge syndrome is represented by the Q squared. Oh, sorry, by the P squared plus 2PQ because it's a dominant disorder. For C, the question refers to the situation presented in part B, and 3,999 in 4,000 individuals is unaffected by DiGeorge syndrome, and that's represented by R because it is not dominant. It's not the dominant allele. The type of plumage found in mallard ducks is described by 3 alleles at a single locus, and then it tells you the dominance pattern, and then the P plus 2PQ plus 2PR. Oh, I messed up. Whatever. So D was actually just R. E was P plus 2PQ plus 2PR because that allele, the MR, is more dominant, so you have to include the heterozygotes that it goes with. F was the human disease. Sickle cell anemia is the result of an SNP. For G, monomorphic is a term that describes a trait that produces a single phenotype in 99% or more of the population. DEAMS is another name for a local population, and the MHC genes in humans are highly polymorphic.
So for question 3 here, we have Marfan syndrome is an autosomal dominant disorder. For the purposes of this problem, we'll assume that the Marfan syndrome affects 1 in 6,000 individuals in the human population. So what is the frequency of the non-disease allele, which is the recessive trait? So I found it a little backwards. I just did 1 over 6,000, which finds the Q value. You can square that and subtract 1, and you'll get the P value. So B, what is the frequency of the Marfan syndrome allele in the human population, which is the Q value that I found of 0.0129. And then what is the frequency of heterozygous individuals of this population? So you just take your P squared and your Q squared and add those up and subtract from 1, and you find this number, which is your 2PQ. And out of a population of 5 million people, how many heterozygotes are expected? You simply multiply that 2PQ by the 5 million, and we got 127,416. So for number 4, Jeffrey Milton and his colleagues found that these genotypes right here at a locus encoding the enzyme peroxidase in ponderosa pine trees grown in Glacier Lake, Colorado. The observed numbers of these genotypes were these guys right here. So A, determine the allele frequencies for the R squared and R cubed alleles. So we've got right here, we have your P squared, Q squared, and the 2PQ, which all add up to 1. And then after a lot of math, which is the chi-square you guys know how to do, you find an x squared value of 7.0949, and we can reject that for lack of Hardy-Weinberg equilibrium.
Okay, so question number 5. Let's see. Okay, so we're trying to find out the frequency of the CCH, which in this case will be the chinchilla dog and also the albino dog. alleles, genotypes, sorry. So for that, we multiply, we square 0.37, and that will give us the chinchilla dog frequency, which will be 13.69%. The other one, we do the 2RS. So we multiply 0.25 with 0.15 by 2, and that will give us about 3.5%. For the last one, we multiply, we square 0.15, and that will give us 2.2%. For question B, among a population of 2,000 dogs, how many would you expect to have the pale and gray phenotype? It'd be heterozygous, so in that case, that will be the CBC alleles. So you multiply 0.075 by 2,000, that'll give us 150. And for C, to find white coat and dark eyes phenotype, and also heterozygous, we will have to multiply 0.23 by 0.25 by 2, that'll give us 0.15, and then do that with the other allele, which will be 0.23 times 0.15 times 2, which will give us 0.069, and we add those two up together and multiply that by 5,000, that'll give us 920. And for the last question, among a population of 15,000 dogs, how many would you expect to have CCH, CB allele, or CC genotypes? So we square 0.15, that'll give us 0.0225, and we add that to 0.185, that should give us 0.2075, and we multiply that by 15,000, and that will give us 3,013.